Well, good afternoon, everyone. It's my pleasure to welcome you to the Wednesday afternoon lecture, which today is to be given by Dr. Ellen Rothenberg from Caltech. This is, I think, going to be a very interesting tour through differentiation and the way in which her laboratory has been working out those lineage pathways, particularly in immune cells that start with hematopoietic stem cells and decide which direction they want to go and what the signals are that determine that kind of outcome, now defined at a level of precision that I don't think most of us would have thought possible even a few years ago. Dr. Rothenberg got her undergraduate degree from Harvard, her PhD in cell biology from MIT, actually in the laboratory of Dr. David Baltimore, somebody you've heard of. After a postdoctoral fellowship at Sloan Kettering and a relatively brief stint as faculty member at the Salk, she landed at Caltech, where she has been since, and exploring this question of lineage differentiation in various ways since then. Having a chance to look over a couple of her recent papers, I know we're in for a treat here in terms of the way in which she has approached this, in things that look like wiring diagrams might even appear on the screen this afternoon. So get ready for that. I've also been led to hear from those who have had the chance to listen to her presentations before that she's not only an outstanding scientist, but an excellent teacher. She's won lots of teaching awards throughout her career. And so this combination of researcher and teacher, while rare, is a combination that I think promises we will hear a very memorable talk this afternoon. And so it gives me great pleasure to ask you to welcome to the podium Dr. Ellen Rothenberg.
Thank you so much. I'd like to thank all of you very, very much, and Dr. Collins and Dr. Schwartz, especially for the great honor of being able to address you this afternoon. And I'm hoping that I'll be able to take you with me on a tour of some of the things that we think we're learning about T-cell development as a prototype of differentiation mechanisms that work to establish cell fate choice, starting from a very specific but very important initiation point, which is starting from stem cell progenitors as opposed to a pre-patterned loaded for action embryo. So I will be talking in great detail, I think, about T-cell development, but I'm hoping that some of the larger scale lessons from this talk will actually apply to a much broader number of cell fates. Now, when we think about definition of cellular identity and differentiation, we're really talking about cell type specific gene expression. This is really what gives a cell its identity. And in order to explain how a cell gets this identity, we have to explain the regulatory state, that is, the combination of transcription factors that are responsible for enabling the cell to express this particular set of genes. That means that when we're asking how a cell acquires its identity, we're really asking two kinds of related questions. One is, what kind of transcription factors make the difference between the cell type you're interested in and its closest relative or its precursor? That is, what changed at the regulatory level that gave the cell its identity? And second, how is a specific regulatory state established from a multipotent precursor? What kinds of regulatory factors and interplays between regulatory factors give the progenitor cell its multipotency, and how are these altered in order to push development down one particular pathway? Now, in many cell types that have been studied very productively, the answer can be parsed into something fairly simple. You can almost approximate cell identity in some cases by the presence or absence of a particular important transcription factor, or maybe a duo of transcription factors. I mean, very early on, the erythroid fate here at NIH was shown to be dominantly regulated by the transcription factor that's now known as GATA1. It was originally called RF1. We've found very productively over the last 10 years that B-cell fate is determined by the lineage-specific combination of EBF1 and PAX5. myeloid fates in a sort of a more interesting and perhaps foreshadowing the rest of the talk, a more subtle way, are regulated by PU1 and CBP alpha. And in a shorthand kind of mental approach, you can think about what makes a B cell as being reducible to the problem of what turns on EBF1 and PAX5. Why? It's because these kinds of lineage-specific dominant regulators are direct positive drivers of lineage-specific genes. Also, very importantly for the developmental issues, they can act as direct antagonists of alternative lineage regulators. Furthermore, to make differentiation unidirectional, they have been shown in virtually all of these cases to be able to act as antagonists of stem and progenitor cell genes, so that once they reach critical levels, the cell cannot go backwards. And what gives these explanations their kind of cogency and their satisfying quality is that the expression of these factors themselves is lineage specific to a large extent, so that once you've turned them on, you can understand that gene will drive, its product will drive the expression of the other genes that are associated with that cell fate in a fairly predictable way. And this is just to say that in order to give a transcription factor these dominant roles, it's important to keep in the back of your mind that one is actually assuming something else about the circuitry, not only about the presence or absence of these distinguishing regulatory factors, but about their relationships to their target genes and to other regulatory genes. So for example, one of these factors can be powerful because it is a direct positive driver of a whole set of genes that are needed by the cell type you care about. In order to make choices, though, as many of you are involved in your own work, you want there to be the ability of the factor you're interested in that drives fate X also to be able to antagonize the activity of factors that drive alternative fates in order to make developmental decisions coherent, in order to make them so that they are simple Boolean-like decisions. And so, for example, if this is another factor that drives fate Y, you want your factor of interest to be able either to block the synthesis of that factor or block its activity, or both. And there are various versions of this that you can do. You can have your factor being working at several different levels of a specification hierarchy cascade. And very often you need to be able to guarantee that your factor of interest will be stable in its own expression if it's going to continue to play these roles. So if it's not being used intermittently, you may want it to be able to positively auto-regulate. And these are all features of many, if not all, of the factors that I just mentioned before. So lockdown is extremely important, especially for hematopoietic cells. And I'm going to argue that T cells are a particularly good model system in which to consider this. The simplest lockdown mechanism to make a cell fate decision permanent is, of course, to program that cell to stop dividing. Because once it stops dividing, it no longer dilutes out its previous complement of regulatory factors. It simply continues to do what it's already been programmed to do. Since T cells continue to grow long after they're specified and carry out their functions with extensive proliferation and extensive mobilization to different microenvironments, you have to have a much more robust type of lockdown mechanism. As I mentioned, these positive regulatory factors, if they're going to be used to determine cell type, they may have to act in positive feedback loops. But furthermore, if you're going to continue to exclude alternative fates and make the cell committed to its identity, you must also do something to keep the other regulatory factors off. You can you silence them in development, but you may have to keep repressing them. And in many, many cases, it's an open question how alternative lineage regulators have this repression maintained. One possibility is by continuous expression of a lineage-specific repressor as part of the regulatory state of the cell. Alternatively, something that many people have been very attracted by is the notion that repression is stabilized by adopting a closed chromatin configuration, which creates a kind of an activation energy barrier for these excluded regulators. And I'm bringing out this kind of background because I think many of us have in mind the notion that if we really understood T cell development, there would be a factor that would be T cellness, or maybe two factors that would be T cellness, and that once we understood their identities and their target genes, we would have solved the whole problem. What I'm going to argue to you today is that that's not true. Instead, what I'm going to say, and this is going to be the end of the talk too, is that in fact T-cell identity emerges from an overlap of a significantly large number of overlapping regulatory functions, none of which is necessary and sufficient, one word, but rather each of which has a different role in this process, some of which are positive drivers of T-cell gene activators, but none of which are sufficient to be T-cell specific all by themselves, and some of which in fact are going to be dedicated repressors, which I'll be talking to you about at the end of the talk. Now, T-cell development is a wonderful system for asking about the pathway from a stem cell to a committed cell, because of the fact that the progenitors have to migrate to the thymus in order to differentiate into a T cell separates them away from progenitors of other blood cell types. And in fact, they separate themselves out at a point when they're still multipotent. And so you can watch, by using a multi-parameter flow cytometry and sorting, you can watch the way the cells move from stage 1 to stage 2 to stage 3 to stage 4, and test the cells, which are portable, for changes in their developmental potential. And therefore, we know that developmental potential is lost not in one step from multipotent to committed, but rather through a sequence of programmed intermediates in which different potentials are lost at different points. So the first ones to go are erythroid and megakaryocytic. As far as we know, these are lost by the cells that go into the thymus no matter what else they have, but many other potentials are still present. What's going to happen next is that the cells will lose the potential to become B cells, and they'll lose the potential to become myeloid cells, while retaining the ability to give rise to other things. You can isolate these cells prospectively and show they still have these potentials and not those. And then finally, the cells become committed. And so the question can then be dissected into a series of problems. What happens between here and here to cause the loss of this fate, but not that fate? And this is crucial because it's saying the issue is not when does the cell acquire T-cell identity, it's through what steps does it lose access to these other identities. And I'm going to try to relate this to the question of how the cells acquire their T-cell identity throughout this talk. Now, this is a diagram, a slightly more anatomical diagram of what the T-cell development stages look like. Progenitors, most likely CCR positive, lymphoid biased, multipotent progenitors, enter the thymus, where they undergo quite a lot of proliferation before they begin really acquiring a definitive set of T-cell characteristics. I'm going to refer to these cells here as ETPs at this first stage. As the cells then begin to differentiate toward the T-cell pathway, they begin to migrate outward through the thymic cortex. These cells are called DN2 cells. I'm going to make a distinction between early and late stage DN2 cells, A and B. Around the time that they reach the periphery of the thymic cortex, they acquire a DN3 phenotype. And this is the time when most of the intense activity in T-cell receptor gene rearrangement reaches its climax. These cells cannot go any further then unless they're successful in T-cell receptor gene rearrangement. They then go through beta or gamma delta selection, which discards the failures. And it's beyond this point, really, that the cells undergo what I would call the immunological part of their development. This is when their fate will be determined by their success at expressing a T-cell receptor, the nature of that T-cell receptor, the types of interactions it has with ligands. Now, from here on, I'm not going to discuss development at all, because by this point, as I'll show you in a moment, the cells already know that they're going to be T cells. And my question is going to be, how does a cell that still has access to these alternative fates determine that the only one it's going to take is to become a T cell? And that happens by this stage. One criterion one can use to determine when the cell decides to become a T cell is when does it activate a T cell specific gene expression program. And this is just some data from real-time quantitative PCR experiments from our lab showing the expression levels of a whole set of different T cell genes on a log scale here. This is a log 10 scale here, and showing how their expression rises during this DN1 or ETP stage, DN2, DN3, and so forth. And in each case, the yellow or green is the DN3 stage. And I think you can see that for the great majority of these T cell specific genes, the peak level is reached perfectly well by the DN3 stage. You don't need to go beyond that, except say for ZAP70, to confer upon the cells the benefit of turning on these genes. So something has happened that distinguishes a cell here which has very little detectable CD3 expression, very little detectable CD3 epsilon expression, to one that's expressing, more than 2 orders of magnitude more CD3 at this time point. So there's positive regulation changes between here and here, which turn on T cell genes. However, the question of commitment is not only the acquisition of characteristics, but the loss of potentials. And let me just take a moment to clarify exactly what I'm going to mean when I talk about alternative lineage potentials and plasticity through this talk. The thymus is an environment which drenches the cells with signals, specifically through the Notch pathway, which we'll talk about in just a moment. And together with signals from particular cytokines such as IL-7 and KIT ligand. And this is obviously an inductive microenvironment. And when I'm talking about plasticity, I'm saying what would happen if I took a cell that was in the thymus in vivo and simply took it out of that environment and put it in a tissue culture dish with something else that would allow another type of cell to grow. What would the cell do then? Would it die? Would it stay a T lineage cell, or would it become something else? And when I say that the cells have plasticity and alternative potentials up to the DN3 stage, I mean that all you need to do is take them out of the dish and feed them other cytokines, and they will happily turn into these other things, dendritic cells and natural killer cells. When I say that DN3 cells are committed, I'm saying that you can take these cells out of the thymus and they will no longer do that. Now, alternative kinds of more heavy perturbation experiments are really hypothetical experiments to get at the mechanism which is causing the distinction between a cell that has potentials and a cell that does not. And this is a case where you could hypothesize that what was really missing was whether the cell expressed a receptor for the growth factors that you were going to let them respond to here. And this is a situation where you're hypothesizing that what's really different is the presence or absence of a particular transcription factor. And in the case of T cells, we know that the cells beyond this point cannot be caused to go back into myeloid fate, say, simply by forcing them to express a growth factor receptor. So it's not simply that they can't sense the presence of things like KIT ligand and FLT3 ligand at these stages, but rather it's that they don't have the transcription factors to execute the program. If you add those transcription factors back, they will then execute that program. So this is not natural plasticity. This is a proof that CEBP alpha in this case, PU1 in this case I'm going to show you in a moment, are sufficient to give the cell back access to a pathway that it had naturally cut off. Okay, so what kind of evidence shows us the cells have changed their access to these pathways? This is just an example of some data from recent experiments by Mary Yui in my lab, fine staging the exact point at which these T cells lose access to the NK and dendritic cell pathways. And I think you can see, even without even reading the axes in any detail, this is the dendritic cell marker CD11C, the NK cell marker NK1.1. Cells from the ETP stage can easily make both. Cells from the DN2A stage can make both, but if you subdivide the DN2 stage into DN2A and DN2B, the cells with a DN2B phenotype have already completely lost access to this pair of fates. And you can quantitate this in terms of the number of dendritic cells per input and K cells per input that you get from each of these stages, ETP, DN2A, DN2B. You can see the fall off, and you can see that it's not just due to a survival issue under the conditions that allow these cells to emerge, because if you add a BCL2 anti-apoptotic transgene, it really doesn't change the breakoff point. So this is simply to say that when I tell you that we know when the cells become committed, it's really a fairly robust phenotype. It's not something subtle. Now, as I told you, this is just a more elaborate version of this diagram, and you can see that the loss of dendritic cell and NK and some myeloid potential occurs just before the cells begin seriously to rearrange T cell receptor genes, almost on top of the branch point between alpha beta T cell development and gamma delta T cell development, for those of you who are interested. But this is long before the later subdivisions of T cell fate that will lead to different types of TH cells, NKT cells, and Tregs. And notice that the loss of B cell potential, again, happens earlier, either just before or just after the cells enter the thymus. So this is a clearly defined difference between the time when B cell potential is lost and the time that these potentials are lost. And this is the period in which the T cell genes are being turned on. Now, what can be driving this two-stage commitment process, and what can be driving the T cell identity acquisition? The dominant signal, as I said to you, from the thymus environment is clearly the provision of delta ligand for the Notch receptors on these developing T cells. And this is a hugely important aspect of the thymic microenvironment. And we'll be using this again and again and again. If you give the cells Notch delta signaling, and many different kinds of multipotent cells can in fact be entrained into the T cell pathway. And I'll show you in just a moment some of the things that Notch signaling can do. But you'll notice that the period of Notch signaling where it's required for T cell development bridges the entire period from the loss of B cell potential through these very separate steps, all the proliferation that occurs in these stages, all the way through to here, and then on through beta selection. And we know that Notch is working at several different levels. We know that Notch delta signaling converts the intracellular domain of the Notch transmembrane molecule into a transcriptional coactivator for a factor which is called RBPJ kappa, or CSL. That's diagrammed here in this little black box at this point, CSL or RBPJ kappa. And the Notch association with this factor converts it from being a repressor to an activator. And so it becomes a direct positive driver of genes which could either be at the level of differentiation genes, part of the T cell program, or at the level of upper level regulatory genes that then activate a cascade of consequences. In work that my lab has done over the past, say, 5 years, we've also found that Notch delta interaction has a number of other more subtle interactions with the factors that are important for access to alternative cell fates. And that there are ways that the spectrum of target genes that these other factors can access can be modulated very strongly by the presence of Notch signaling, and the molecular mechanism for this is still being studied. So Notch is clearly important, but for our purposes here, I just want to point out that there are limits to what Notch signaling can explain. If you actually do an experiment in which you ask, how much does the expression of such and such a gene change between the ETP stage and the DN3 stage, that is the beginning of the intracellular differentiation process and the commitment stage, and you ask, is the degree to which those genes get upregulated or downregulated a direct function of their sensitivity to Notch signaling differences? The answer is No. This is actually a plot of this kind of data right here, where the little yellow and turquoise triangles are data from 2 totally independent experiments in which we were just looking at the levels of these genes in thymocyte subsets, and this is the ratio of expression in the DN3 stage to the ETP stage, from the highest to the lowest. And the red bars are the extent to which the expression of those genes depends on Notch signaling. And you can see, yes, there are a couple of things which are upregulated at DN3, which are also Notch dependent, like deltex, like pre-T cell receptor alpha, but there's an awful lot of disparity between these curves. So that what that's telling you is obviously you need additional players. Yes, Notch is one of the inputs, but you're also going to need other regulatory inputs. So what are they? In order to try to find out, over the last few years, my lab has been trying to get various ways of doing a census of regulatory factors that are expressed during these early stages of T cell development and looking at their expression patterns. And this is just one of the figures from one of these gene discovery papers that was published about a year ago, in which I'm just showing you the expression of a number of different transcription factor genes. Some of them are being assayed in 2 separate experiments, but where they've been clustered to sort of separate those that have a rising pattern of expression, a falling pattern of expression, and a number of other patterns, some of which have a very sharp peak at the DN3 stage, some of which, very interestingly, are expressed at unchanging levels from the multipotent progenitor stage all the way through commitment and only change expression afterwards. And so there are clearly a lot of players, and there are quite a lot of potential regulatory changes, both in terms of gain of function that may be T cell specific, and very interestingly in terms of losses of function. I think an important thing to bear in mind, especially when trying to understand the multipotentiality that continues in the cells until commitment, is that the point of departure is a continuing expression of a number of stem or progenitor cell transcription factors, actually quite a very large set, at levels that are quantitatively hardly changed at all from pre-thymic precursors. And this means that there's an enormous amount of continuity that the cells are maintaining with their stem and multipotent progenitor forebears throughout the period when they are going from no T cell gene expression to full T cell gene expression and multipotency to complete commitment. Now, some of the stem and progenitor genes also play a role in alternative fates, and we'll talk about PU1 in just a second. And these are then a direct pathway through which being a progenitor cell can also be a door towards a particular non-T alternative. But this is the background against which the T lineage specific positive functions have to be activated. It is anything but a blank slate. It's actually a quite complex system, and in a sense, the T cell program is going to be established in competition with a prior progenitor cell program. So there's actually relatively few regulatory genes that seem to make the difference between the committed cell and the multipotent cell. And this has been slightly extended by our more recent whole genome analyses, but actually it's been extended a lot less than we expected. And so it's, these are still a really good set of genes to use to score this progression to commitment. One is that a set of DN3 specific genes goes on as the cells enter this committed state, and the other is that there are 3 tiers of stem and progenitor genes that go off, 2 of which are controlling access to different kinds of growth signals, several of which are transcription factors that are involved in aspects of multipotency and alternative lineages, and then finally a set of progenitor cell genes that go on only at the completion of commitment. And this is just to show you what some of these losses of stem cell gene expression look like, because they're actually quite important for what I'm going to tell you about for the rest of the talk. And some really good examples are LYL1 here and PU1 here. Notice that these are log scales. These are quantitative RT-PCR measurements. And what you're seeing is that as the stars are indicating the point when the cells change from being biologically testable as uncommitted to being biologically testable as committed. And you can see that each of these genes has its own pattern of downregulation, but say GFI1b, although it's not expressed at a high level initially, falls off a cliff during commitment. PU1, a very important hematopoietic regulator, decreases a little bit, then decreases another 5-fold, and then falls off a cliff. LYL1 is a very beautiful example of this pattern because it stays at a fairly high level until just the point the cells are committed, and then it goes off, and so forth. And so these are large-scale changes in gene expression, something like 100-fold over the whole course of this transition. This is just to show you by intracellular staining for PU1 that these changes in RNA expression really do get reflected in changes in protein expression. And what you can see here, the blue and the red curves show the levels of intracellular PU1 staining in the ETPs and the DN2s that are not committed yet. This is the green curve here, shows the level in the first committed stage, and you can see that it's perceptibly shifted, and by the DN3 stage, it's substantially down. And this is showing that the loss of PU1 is at the protein and the RNA level correlated with commitment. Now, I said I would say something about PU1, and we spent a number of years really focusing on PU1 because it's well established not only to have a role in multipotent progenitor cell generation, but also as a very active participant in access to the myeloid and the B-cell programs. And so it seemed like a one-stop shopping for an explanation of some of the losses of potential that the cells undergo. And indeed, you can do a kind of a Cox postulate experiment. You can take DN3 cells that have turned off PU1 already, add PU1 back into them, and then what happens to them? Well, it turns out that if these are your little DN3 thymocytes under the control conditions, if you add back PU1, you get large cells. They're MAC1 positive. You can see they're not lymphocytes at all anymore. You start to see some nuclear segmentation occurring. If you put this into fetal thymocytes and grow them for a few days in the absence of Notch signaling, you can see this wholesale change in phenotype with the cells acquiring MAC1 at a high level, F480, and low levels of CD11C and so forth. So the cells have really just in a few days, they completely change identity if you add PU1 back to them at a point even after they had turned it off. So it's sufficient to restore and force access to this other fate. We thought that PU1 might have a more interesting role as well, though, because of the fact that it is naturally a part of the expression profile of early developing T cells. And we were very interested in the possibility that its expression early on might be part of the stem-like expansion of the cells before they undergo T cell receptor gene rearrangement. And one way to look at this was to ask if we could make sense out of the pattern of genes which were affected by overexpression of PU1 and relate them to the normal trajectories of gene expression through the DN1 to DN3 period. And this is a sort of a principal component analysis done by my collaborators, Konstantin Georgescu and Hamid Balouri, in which we attempted to relate the expression of about 146 regulatory genes from the study that I showed you before to principal components that really explained most of the change in gene expression during the period from DN1 to DN3 and all the way through beta selection. And it turned out that actually the first principal component is a pretty good approximation to the DN1 to DN3 transition. This is where most of the change in regulatory gene expression occurs. And it turned out that if we look at this and superimpose on this the effects of PU1 on these genes, we find that if red is a stop sign for expression of these genes and green is a go sign for expression of these genes, we see that PU1, which itself is over here in the DN1 side, is in fact a very good negative regulator of a whole slew of genes that are involved in this DN3 stage, while it's a positive regulator of some that are involved in the DN1 and 2 stage. So PU1's own pattern of target genes, even within the context of T-cell development, could be playing a kind of a rate-setting function in the progression of the cells towards this DN3 stage. However, so PU1 obviously is something the cells will get rid of at an early stage. And so we felt that the question was, is there a positive regulator of T-cellness that we could look for and to understand its effects in the acquisition of the climax T-cell identity? And there is a problem here, and this is a diagram in which I'm basically trying to summarize a lot of research from many labs in the literature. There's no shortage of regulatory genes that are required for T-cell development. PU1 itself is required for T-cell development, but there are many others. The only problem with them is that none of these, as essential as they are, none of these is T-cell specific. And this is a huge contrast with the case of EBF1 and PAX5, and even in a more restrained sense, it's a contrast with the use of GATA1, for example, in the erythroid megakaryocytic lineages and so forth. Because not only do these factors play a role in T-cell development, they actually are critical positive regulators of other fates. For example, E proteins, which are required by T-cells, are the top of the cascade for B-cell development. GFI1, critical for T-cells, critical also for neutrophil development, and so forth. And so we're forced to a situation where T-cells have to be seen as a mosaic of inputs from several factors, none of which are T-cell specific to date, and all of which have roles in actively promoting alternative cell fates. Let's take the best case of a T-cell specific regulator, and I'll show you that even it is not T-cell specific. GATA3 was thought to be more or less T-cell specific until fairly recently. And so the question was, is GATA3 the factor which positively confers T-cell identity? And in fact, GATA3 expression rises between the prethymic stages and the ETP DN2, DN3 stage and so forth, and then it stays on to play roles throughout T-cell development, quite in contrast to PU1. So, what is it doing? Well, if you add more GATA3 to cells that are going into the T-cell pathway, the interesting thing is it does not make more T-cells. It does not help them become T-cells. It radically blocks them from becoming T-cells, in fact. And the only way they can live with more GATA3 is to become mast cells, and they're beautiful mast cells that develop out of thymocytes if you add more GATA3 than they were expressing normally. This is not an ectopic factor. This is just a dose effect. And so even GATA3, which has a relatively T-cell specific expression pattern, is not T-cell specific in its effects downstream. And so this is a kind of an example. This was one of the best cases for a factor that might have been expected to give T-cell specific effects, and it really doesn't. So lessons from these. The PU1 effects are modulated in the presence of notch signals, which I alluded to. But even T-cells, but even so, it is trying to force the cells into another pathway or backward toward a stem cell-like state. The GATA3 effects are very dose-dependent. They are not T-cell specific unless they're at a low dose. And all of the effects can only be brought under control by another event, which is what I'm going to talk about for the last part of this talk, and that is what closes the window of susceptibility to alternative lineage fates. First, just to say that we have attempted to figure out how the known perturbation effects of the factors that have been studied so far can map onto a program of T-cell development. And this is just an example of a draft that we've published in which we've tried to organize data from our own lab and data from other labs to try to combine the levels of accountability that exist in the literature so far for the genes whose expression we know has to change in the progression from multipotency to commitment. In this diagram, the stem-like genes and erythroid genes are over here, B-cell genes here, myeloid genes here. These are pre-T-cell genes, and these are going to be involved in beta selection more or less, and some NK genes are down here. What I want to point out is that this is a very incomplete model. If you actually go to our website, you can find this is it's incomplete partly because it has too many lines on it. What we have is perturbation effects from a relatively small number of experiments where we've been able to play with the levels of PU1, GATA3, Notch signaling, and so forth. We haven't been able to play with the levels of everything in here. Therefore, when I say that there's an effect of PU1 on HES1, I don't know whether it's going directly or whether it's going through something else. And it's very possible that when we fully understand the something else's, we will be able to simplify a number of these parallel lines going through here. And I would want any of you, I'd like to invite any of you who are interested to check out the website where this is live. It's in a program called Biotapestry. You can mouse over any of the links and it'll tell you what the evidence is for that particular linkage. And this is meant to be a work in progress that we're trying to get going. But what I'd like to point out just as an entrance to this last part is that there were several functions that we could not account for. One of them is a DN3 specific positive regulator, which I told you it is not GATA3. The other, though, is a negative regulator that accounts for the closing off of the stem and progenitor program. And that's what I'd like to spend the rest of the talk on. So commitment is about repression. Commitment is about loss of access. And so the real question is there one master commitment factor that's a master repressor for all alternatives, or are different mechanisms required? Second, are the repression events really separate from the activation events? Now, this seems obvious, but in fact, in B-cell development, both PAX5 is certainly shown to be a bifunctional transcription factor that can both participate directly in activation of B-cell genes and participate in a rate-limiting way in the repression of non-B-cell genes. So are we looking for one factor or many? And when do these events occur? Are they hierarchical? Can we actually map the changes in expression of the usual suspects for each of these alternative fates onto the timing of the loss of potential? So to get at this issue, my lab has been, one of the things that we've been doing is start to look at some whole genome analyses of histone modifications across this interval from the uncommitted, basically ETP stage, to the committed DN3 stage. And we've been looking at basically 3 marks across these stages. For activation marks at particular cis-regulatory sites, we've been using acetylated histone H3. For accessibility separate from activation, we've been using H3K4 dimethylation. For repression, we've been using K27 trimethylation. And this is being carried out as a collaboration with Barbara Wold's lab, and specifically with Ali Mortazavi, who's been helping us. And the student in the lab who's been doing this is Jingli Zhang. And I just want to show you a tiny bit of this data just to show you that you can start to answer some questions immediately this way. First, just to orient you, the top rows are going to be acetylated histone. They're activation marks. And you can see that when you have a gene set like the CD3 genes, these are not expressed in the DN1 stage very significantly. And you see that there's very, very little acetylation here. There's actually a little bit of K27 trimethylation, in fact. But you can already see these dimethylated K4 islands. And these are going to serve as nucleation sites for a broadening of these activating modifications when you go to the next stage, when the gene begins to turn on. And this is just an example of how they go on while this tiny little possible hint of K27 trimethylation goes off. Very different picture is seen as you look at a B cell gene, PAX5 itself. This is going to be repressed from the very beginning of this process. And indeed, you see a very thick fuzz of K27 trimethylation covering this gene, not only when the cell's committed, but really from the stage when the B cell potential is lost. At the very, very beginning. You can see there's absolutely no activating mark here. This is the promoter of the next gene over, ZCCHC7. This is PAX5 itself. But what you can see is that there is still K4 dimethylation in beautifully defined locations that include the documented enhancer of this gene, so that you can still see where the regulatory sites would have been if the gene had been active. And in fact, this kind of early repression affects the B lineage-specific regulators and some myeloid-specific regulators, including MAFB as well as CEBP alpha, and the stem cell mast cell regulator GATA2. Now, in contrast, we have some of these stem-like genes. I pointed out LYL1 to you before, and I told you that it's expressed initially at a quite high level, but then it's repressed. And you can see that very nicely here. Here's LYL1. It's this little tiny guy in between two other genes. Here's the arrow that shows where it is. It's very strongly activated in the DN1s. As the cells go forward, it's still quite active, but by the time you get to the DN3 stage, this activation mark is gone, and you've now started to see what is the beginning of a focus of repression, which is going to spread at the next stage, which I don't show you, across the gene. And so this is one form of DN3 stage specific repression, which involves the mobilization of the polycomb PRC2 complex for methylating K27. This is one way to shut off stem cell genes. However, it's not the only way to shut off stem cell genes. And an example of this is PU1 itself, the SFPI1 gene, which starts out again brilliantly on. That's the acetylation. That's the dimethylated K4, but it's going to start to fade away at the DN2 stage. It will be gone here. And what is interesting about this is you still see absolutely no increase in many experiments beyond these here. There's no increase in K27 trimethylation. So it's being repressed at a similar stage to LYL1, but through a different mechanism. And so what this has already told us is that there are at least 3 different kinds of repression being lavished on these cells. One which is early repression, one which is late repression involved with the PRC2, and one which is late repression through another mechanism. Okay, there's actually a third cohort that will be involved. So do we have any clues as to what the nature of these repressors might be? And this is a relatively recent set of results that have just come out, but we're very excited about them because we think that we have a handle on at least one of the major repression molecules, and that's this last bit. You remember that GATA3 was inadequate as a DN3 master regulator, and what I didn't tell you is it doesn't even have the right repression functions. However, we have a new candidate, which is a tumor suppressor gene, BCL11B, which is extraordinarily T-cell specific in its expression beyond anything that anyone has really described before, and it turns out to be a very good candidate for this. So this is just an example of when BCL11B goes on. It goes on between DN2 and DN3 very dramatically. If you between DN1 and DN2, I beg your pardon. And if you ask when it goes on, it actually goes on just before commitment. It goes on between DN1 and the DN2A stage. It rises to its peak at DN2B. And although it's not shown here, it stays on then for the rest of T-cell development. And it's not expressed, as far as we know, in any other hematopoietic cell types. So the question is it doing anything in early T-cell development? And we've been very fortunate to have the opportunity to address this through a very kind collaboration with Dr. Mark Lead from Oregon State University, who had a LOX-P tagged BCL11B allele, and he was willing to give us the mice. And in this gene, virtually all of the 6 zinc fingers that are involved in DNA binding by BCL11B are encoded by exon 4 of the gene. And so this was easy for him to flox. And so what we've done to look at its effect is not to look at it in knockout mice, but to take these floxed cells and to allow them to isolate what are still completely normal hematopoietic stem cells, and then infect them with a Cre retrovirus and ask them to develop into T cells in vitro so that we can follow the fate of a cohort of cells, and we can actually take the cells out partway during the process and assay them again. And for this, we're using the famous OP9 delta culture system that was pioneered by Schmidt and Zuniga-Flücker, who's, of course, a former product of this wonderful institution. And this is really what I would consider one of the major breakthroughs in T-cell development that's made it possible for people to take multipotent progenitors and create committed T-cell progenitors in vitro completely. And we've been able to then follow the fate of these cells over the next weeks. And the first thing that you can see is that there's a commitment defect in these cells, because loss of BCL11B makes it much more easy for these cells to become NK cells. And you can sort the cells already at DN2 stage, and they love to make NK cells. BCL11B is known to be required for development to the double positive stage through beta selection. But we're now looking much earlier than beta selection. We're looking at the acquisition of T lineage identity itself. And what you can see is that these cells will go on to keep this NK or IKDC, actually developmental potential, even after they appear to have acquired a phenotype that might be otherwise viewed as a committed phenotype. So this is very interesting. And we've actually asked what happens to these cells. And what you can see is that if you follow the development of these cells to the DN2 stage, which they do just fine without BCL11B, and then you sort the cells that are DN2 and ask, well, will they go on from there? If you take them from this point from control mice, they will go on to give rise to later stage cells. That's how you know that a DN2 stage cell is actually an intermediate in T cell development. But if you take them from the BCL11B conditional knockout mice after they've been Cre-treated, they just give more DN2A cells and more NK-like cells. And this is just shown here. These are cells that were sorted as DN2A or DN2B cells from a B6 control or from the BCL11B in vitro knockout cells. And what you can see is that whereas the DN2As and Bs from the B6 mice go on to give rise to sort of DN3 and DN4 cells, the ones from the BCL11B floxed ones are stopped here. What's more important and what's more interesting is why they're stopped, at least why we think they're stopped. And I'm going to skip this for a second. And the answer is because they cannot shut off the stem cell phenotype at the molecular level. And that's what I'm going to show you here. So this is taking the cells that I was just showing you that are that either go, this would be sort of where the NK-like cells are. DN1 cells are cleared out. True ETPs are cleared out. But there's 1, 2, 3, 4. And so what we're going to do is we're going to take these sorted cells and then ask what they can give rise to and what happens to their gene expression pattern. And these are the populations that we're going to isolate. And in this final slide, the blue bars show the pattern of expression of genes in the B6 control cells. They're normal, and you'll see there'll be BCL11B itself will be upregulated and then it'll stay high. T lineage genes will be upregulated and stay high. And the brown bars will be the BCL11B deleted cells. And there are some interesting features of this pattern. First, this shows that BCL11B was indeed deleted in this experiment. This will show you that the T lineage genes can be activated initially to their DN2 levels in the absence of BCL11B. So it's not needed for the initial turning on of T cell specific genes. They don't go up to their maximum level, but they start to go on. However, the huge problem with the BCL11B knockout cells is shown in these bars out here, which is that they fail to turn off PU1. They fail to turn off LIL1. They fail to turn off SCL. They fail to turn off BCL11B, GFI1B. Basically, they fail to turn off that stem cell legacy set of factors which have to go off at this point. And so these cells are very, very happy. They will grow for weeks in culture. They are not dying out. They are perhaps even happier because they continue to express these genes. But without BCL11B, they don't have the repression mechanism that they need to silence them and therefore allow the cell to fully complete its commitment to being a T cell. So finally, what I'm saying here then is that among these various layers of genes, we have stem multipotency genes which make it possible to turn on some of the T cell program. There are some alternative lineage genes that will have to be silenced in this process. Some of them may be silenced quite early, but there's a big problem with silencing the stem and multipotency genes, and that one of the genes that has to be turned on in order for this job to be executed correctly is a product of a T lineage specific activation machinery, BCL11B, which then plays a very important role in turning off at least some aspects of the stem cell multipotency program. And we think that one of the major take-home lessons of this system is not that BCL11B is the factor that we've all been looking for that is the master regulator of T cell development, but that T cell development reveals the need for layers of regulatory action, complementary and quantitatively precisely balanced layers of regulatory action that are required to guide the cells with extensive proliferation through an ordered, temporally regulated sequence of steps safely to commitment, but with the acquisition of the right balance of factors to turn on the T cell genes without killing the cells, without diverting them. And we think that this T cells are perhaps a very transparent system in which to see this because of the ability to isolate these intermediate stages. But if you haven't found a master regulator for your cell type yet, it's worth considering that a similar kind of layered structure is involved in the specification of the cells that you may be most interested in your own research. So I'll stop there, and I'll just mention the people who did the work I told you about. The work on the fine-tuning of commitment was mostly done by Mary Yui with Ni Feng, a very good technician. The work on the whole genome analysis of the epigenetic changes associated with T cell specification was work of Jingli Zhang, together with Barbara Wold's group, Ali Mortazavi especially, and the facility that's run by Igor Antashechkin. I'd like to comment on the work by Deirdre Scripture Adams, Chris Franco, and Tom Tagon beforehand on the effects of PU1 and GATA3 on the cells and the role of Notch in trying to keep the cells on the straight and narrow and punishing those that try to divert from the T cell path. And finally, Long Li, together with Mark Lead from OSU, have been extremely important in our emerging understanding of the role of BCL11B. Thank you very much. Yeah, and I should have mentioned Constantine Georgescu, Bill Longabaugh, and Hamid Balouri for the computational work. Yeah, thank you. If there's time, I'd love to take questions.
By all means. That was a fascinating travelogue through a whole complicated set of pathways, but I'm sure many questions come to mind. The microphones are in the aisles as usual, and please feel free to put forward questions to Dr. Rothenberg, beginning with Dr. O'Shea.
Ellen, in looking at more mature T cells, our lab with Bill's and KG Zhao, we found that in contrast to the cytokine genes, which really acted like their epigenetic marks, when they're off, when they're on, the so-called master regulators really seem to have a fear of commitment. You know, they had these bivalent marks, both K4 and K27. And I was just wondering, for all of your so-called, master regulators, the T cells, do they also have bivalent domains? Do T cells really, are they afraid of commitment?
Well, you're talking about a fine aspect of commitment, right? Because your cells are not stopping being T cells. No, actually, I think I'm getting a slightly different view of the role of these marks. For example, BCL11B itself starts out completely plastered with K27 trimethylation in this period, and it has no trouble being activated once the time is right. So I think that it's not, now that is really why it has such a steep activation profile, because it's really well silenced before, but it can be turned on. So I think that my perspective in this is that the jury is really still out on whether there is any generalization here. We clearly have a mechanism for PU1 repression, which I didn't talk about today, that does not involve the installation of repressive marks. At the same time, we have genes like ETS2, for example, which starts out looking like an active gene, except there are no transcripts coming off of it. And so I think there's going to be a group of different mechanisms that are used on a gene-specific basis that my guess is they'll reflect the biochemical mechanisms of action of the particular transcription factors that are either promoting them or holding them in check. Yeah.
So, Ellen, it used to be considered that B cells and T cells were directly diverging from a common precursor, and now there is another perspective that, in fact, the divergence of B cells would precede and would actually diverge from a myeloid and T cell common precursors. Now, one thing that B cells and T cells have in common is the necessity to turn on RAG. So in your studies, have you ever seen a commonality in this gene expression program that would turn on RAG expression in B cells and T cells, or have they derived completely different programs to do that?
I think that in the early double negative cells, they may very well be using a similar mechanism, and the main thing about RAG expression is the E protein dependence. So this is combinatoriality. So if you have a gene which responds to a factor which is common to both programs and which is not prohibited by a factor that's in one of the programs, you can use a similar mechanism for that. And I think the shared use of E proteins is very big. Double positive cells switch to using a different cis-regulatory module for RAG expression than double negative cells do. And I think I'd still really like to see people go back and look with some care at the element that's being used in the double negatives, because it may be the same as the one in the B cells. And it certainly is going to be, it can be explained quite comfortably by the expression pattern of the activity profile of E2A and HEB, for sure. Yeah, KG. Oh.
Well, congratulations for telling us the biotapestry and designing the whole system, including about 130 genes for the normal development and differentiation from hematopoietic stem cell to T cell. So what goes wrong in this pathway that generates in T cell leukemia?
Practically, so this gives us target genes, but I think one of the things that strikes me about leukemia is the way it uses genes which are absolutely native to the T cell program, and it just makes them miss their appointments with repression. So, or anticipate them. And so it's, I think this is one of the most dramatic aspects of why the repression is important. It not only gives the cell the chance to go from one stage to the other, but the penalty for this is often leukemia. I mean, the cutoff of Notch signaling after beta selection is very important for that. But also, if you have a failure to downregulate Tal1, of course you get leukemia. It's presumably due to a squelching of some E protein effects at the DN3 stage, and so I think that I suspect that LYL1 is working probably through the same targets. So I think that these are native parts of the T cell program that have a natural repression mechanism which can be overridden only at peril. And it's interesting that BCL11B is needed for these repression events, and it is a tumor suppressor. So I think there's a cluster of mechanisms involved, but there are many recurrent hits on things that affect E protein activity, that affect Notch activity, that affect Ikaros activity, and these are all very, very dynamically regulated.
And this work is done with the mouse cells and...
Yeah. This is all done with mouse cells. I mean, it's a tremendous advantage to be able to work with them for many technical reasons. But in principle, a lot of this could be done with human cells.
Thank you.
Take 2 more questions.
Ellen, most of the talk you concentrated on a qualitative assessment of which factors are important for particular processes. But in the course of this, you were honest to say, well, gee, if you change the level of GATA3, then you get a different fate. And so for people who are interested in thinking about engineering cells to a certain fate, as the gentleman to your left standing there, that seems to raise a message about how do we go about thinking of this where these small differences, 2-, 3-fold, less even, begin to change the behavior in the system in these rather gross ways where our qualitative maps, rather than quantitative models, are misleading in a certain way, that the system is just sort of doing its thing and you ignore those numbers until you mess with them, and then all of a sudden lots of things break loose. I won't use the word for that.
Yeah, so I think that it remains to be seen how general a problem this is. It certainly is the case that the factors that we have played with in T-cell development are all incompatible with T-cell development if you go too high or too low. It's very easy to block T-cell development. It's very hard to push it. And even if, in a sense, if you think about Notch, that's the exception that proves the rule, because that's the only way you can get a transcriptional input where you're not changing the actual protein to DNA ratio of the transcription factor. You're simply changing whether it's got an activation or a repression switch attached to it. So it's a very interesting problem about the evolution of the T-cell program. But I think one of the points that comes out of this is that the sensitivity of this early T-cell programming, say to GATA3 changes, goes along with a particular network structure at this stage. There is no GATA3 to GATA3 positive feedback at this stage. In Th2 development, you add GATA3 exogenously and it turns on endogenous GATA3. That does not happen at this stage, and we suspect that the cis-regulatory elements that mediate that are actually buried in heterochromatin at this stage. So what I think that there's something about the T-cell program which may be kind of a worst case scenario from this point of view. Certainly later on you can drive a system by exploiting a transcription factor that's designed to be able to work with positive autoregulatory feedback. And that is so the reason we see these problems here is because that's not the way these are wired up. So is that an answer to, I mean, I think what I was saying to Dr. Collins beforehand is this really reveals the fact that I think the field needs better tools for controlling temporally restricted quantitative control of different regulatory factors. I think it's a major piece of technology that needs to be developed better. But we certainly can sort of come in on it the way the ancient Greek mathematicians figured out the value of pi by polygons around the circle. Yeah. So anyway.
Final question.
So it's clear that one transcription factor may not be sufficient to induce T-cell development, so it's very complex. But I don't think they will be more complex than turning a terminal differentiated cells into stem cells, which only requires 4 transcription factors. So would you speculate on actually how many, maybe 2 or 3 factors will be needed for T-cell development, and do you have a good candidate like GATA3 combination with BCL11B or things like that?
Well, we know that they need BHLH factors. We know that they need ETS factors. We know they need GATA factors. We know they need MIB. We know they need TCF Wnt factors. They certainly need GFI1 at the beginning. I mean, you can certainly, and Runx factors are critical. So, I mean, certainly there's a lot. Now, one of the things that makes it complicated is that T cells happen to use a lot of the redundancy in the mammalian genome for different factor family members. And so that I think that you can probably cut it down to a number of families. But
So has anybody tried a Yamanaka experiment where you just throw the whole batch of factors at
Yeah,
a
well,
fibroblast and see if it goes where
becomes a
you
T
want
cell.
it to with an
Yes.
appropriate selection, and then you leave one out and find out what's really necessary.
yeah, I think it would be fun, but so I think the answer is it's really hard to tell. I don't think it's infinite. I agree with you. I think that we can see recurrent themes in these cis-regulatory elements. There are motifs which seem to be overrepresented. They're the usual suspects. So I think, it's not an unsolvable problem. Thank you.
Well, let us thank our speaker again for a wonderful Wednesday afternoon lecture. And the reception will now be held in the library, so if you have further conversations you'd like to have with Dr. Rothenberg, please come for cookies and refreshments.